CYC is a free channel, presents the Word of God for everyone. Your support will help us to continue the mission. Visit our website, cycnow.com. Even $1 will make a difference.
Welcome to our next session on evolution and science, where we've been talking about science and the Bible, whether they're friends or foes. We've been talking about Genesis 1 and science, and in these sessions we've been looking at the origin of life. Where did life come from? And does the Bible have anything to say about that? And does science have anything to say about that? And how do they fit together? And we've talked about the timing of early life. You know, when did it start? We looked at the conditions of early life. And in this session, we're going to start off with the complexity of early life. That's going to be the focus. But before we do that, let's just introduce our guests today. We have Miriam and Meryl and Eva and Monica with us. So why don't you tell us where you're from?
I'm from the Church of Virgin Mary and St. Athanasius from Mississauga.
Same here. I too am from the Church of Virgin Mary. Me too.
Okay, so they're all from the same church in Mississauga. That's wonderful. So my name's Don Waller, and I'm your host for these sessions. I work with Reasons to Believe, a science and faith organization seeking to integrate science and faith issues. So, the complexity of early life. Let's jump into this subject today. And remember, write down your questions, and we'll look at those at the end. So, when we look at the fossil record, we see a number of interesting things about it. Remember that in our model, our naturalistic model of where life came from, we expected that early life would be simple and that it would gradually increase in complexity. But what we actually see in the fossil record is a very complex ecosystem in the fossil record. We see photosynthesis, for example, having taken place. We see methanogenesis, which is generation of methane, and sulfate reductions and other fancy chemical reactions that a lot of these early bacteria, what are known as cyanobacteria, were capable of doing from the day that they seemed to appear. It's not that they acquired this ability to do photosynthesis, they just had it right from the get-go. We see energy generation and self-replication pathways, metabolic pathways, complex metabolic pathways. I'm a biochemist, and so I grew up in my training staring at charts like this one, which is basically a giant metabolism chart. Explains all the interconnectivity of what goes on inside of a cell, and it's really, really complex. And so this is typical of what some of these early cells were capable of doing. Now here's a quote from a paleontologist who said this: "No one has publicly disagreed with my interpretation of the apex fossils." This is a certain type of, a certain area of fossil discovery. "But privately, some would prefer I were mistaken, since they, and I too, would prefer a simpler evolutionary story. One that told us these oldest fossil organisms were capable only of primitive ways of living, and that advanced metabolic lifestyles evolved much later. But the evidence seems strong, and what one might prefer shouldn't matter." And so at least we have someone here who's honest enough to say, you know, based on what we think the fossils should look like, they don't look like that. And that's really troubling. That's what this guy's saying. He's saying, you know, we expected it to look like it was really simple, but when we look at the fossil record, it's, wow, they're really complex, their abilities, these cells and these primitive organisms. You know, it's like, how do we explain this? And you can see this whole thing of, well, I'm coming from a particular worldview. My worldview is that it had to happen by evolutionary and natural processes. They couldn't possibly have had any intervention by a divine being. And so that really is a problem for this person. Okay, we'll see more of this as we go along. What about that primordial soup? Okay, we talked about the primordial soup. So we're going to look at that a little bit. Here's a quote from Noam Lahab, who is a famous origin of life researcher, and he said this. He said, "So far, no geochemical evidence for the existence of a prebiotic soup has been published. Indeed, a number of scientists have challenged the prebiotic soup concept, noting that even if it existed, the concentration of organic building blocks in it would have been too small to be meaningful for prebiotic evolution. What's he saying? He's saying that the whole of this origin of life thing depends on the earth being covered with this primordial soup, and that somehow out of all that, this complexity of life emerged. But everywhere we look, we don't really see the evidence for it. So that doesn't make sense. Well, it's interesting because in the 1950s, there was a series of experiments done that were essentially designed to try and replicate this early Earth, okay? I don't know if you've ever heard of this, but it's a famous experiment called the Miller-Urey experiment, okay? And basically what they did is they constructed this fancy-looking apparatus with some glassware, where in the one thing at the bottom they had water and the ability to heat it up. And at the top, they filled it with gases that they thought were there in the primordial earth. So they had some methane, and they had some ammonia, they had some water vapor, some hydrogen, and a whole bunch of gases, which they thought is what the earth was like at that time. We're talking billions of years ago, right? In the primordial earth. And what they did was they inserted some electrodes into this glass container where these gases were, and they thought, okay, so we're going to replicate the early earth with all the lightning and everything, and we'll zap these gases with these electrodes and see what we get. And so that's exactly what they did. And what happened was whatever formed came down the tubes and went through a condenser, and they were able to isolate those out, take them out of the experiment. Now, when they did this, okay, the amazing thing is that when they zapped this gaseous mixture with these electrodes, they actually created some amino acids. Now, what are amino acids? Amino acids are the building blocks for proteins. So everybody around the world all went, wow, this is fantastic. Look at this. The results of this experiment shows that, you know, it's possible that life could have happened just with a bunch of lightning and a bunch of gases. That's all we needed. We have amino acids after all, and everybody knows that amino acids are the building blocks of proteins, which are necessary for building cells and so forth and so on. And everybody got carried away with this idea. Several problems with the experiment. The first thing is that the gases that they used didn't represent early Earth's atmosphere at all. They today believe that the atmosphere was quite different and would have made it much more difficult for those to form in that way. And the second thing, and the most important thing I think about this experiment, is that yes, they made these amino acids, but they had to physically take them out of the system before they got back down into the bottom and were recycled again. Because if they hit the water at the bottom, they were instantly degraded. So in other words, the experiment itself would have destroyed those molecules under those conditions if the scientists didn't take them out before it happened. So in other words, they had to intervene in order to get those molecules out. You with me? Mm-hmm. And so basically, the whole thing was set up, you know, to be to the advantage of the researchers. And if it really was replicating the early Earth, and they really did have these discharges and these molecules were created, they would have landed back in the primordial soup and instantly degraded. And so it couldn't have happened. So that's the problem. So here's another quote from some origin of life researchers. Now remember, these are guys that are serious scientists. They're seriously trying to understand how life happened on planet Earth. But is the prebiotic soup theory a reasonable explanation for the emergence of life? Contemporary geochemists tend to doubt that the primitive atmosphere had the highly reducing composition used by Miller in 1953. So what's he saying? He's just saying, you know what, that wasn't the right mix. Now, the next couple of slides here, I just want to show you some of the things that are actually needed in order to have life. Now, I'm going to go through this really fast because the main idea is I just want you to see that it's not straightforward. When you say you want to have a number of things happen in order for life to happen, here's just a few steps. You have to have the assembly of boundary membranes. Okay, you all know that a cell is surrounded by a membrane, right? It doesn't just float around in its environment. It has to have something that encloses and keeps all the insides inside and all the outsides outside, right? Okay, so you have to have those membranes somehow come together and form. You have to have the formation of energy of energy capturing ability of that membrane. In other words, it has to have some way of capturing energy. You have to encapsulate macromolecules within this membrane. In other words, you have to be able to keep all the fancy biomolecules like DNA and RNA and proteins inside the membrane. You have to have transport pores across the membrane. They have to form as well because stuff has to be able to come in and out, but in a controlled way in a cell. You have to have systems for the macromolecules to grow. You have to have catalysts that speed things up, because if everything just worked by normal chemistry, it would be way too slow. So we have to have all kinds of fancy biomolecules called enzymes that make it all go faster. You have to have the ability for these macromolecules to replicate or duplicate themselves. You have to be able to introduce information into these molecules, because they're going to dictate what happens in the cell. You've heard of DNA. Well, DNA is basically full of information. We're going to talk about that a little bit later. You have to have mechanisms that allow this boundary membrane to subdivide so that the whole cell can grow into two cells, right? And you have to have the production of the means to pass information from the parent molecule to the daughter molecule when the cell subdivides. Otherwise, the cells will die. So here's what one guy said about this list. He said, looking down this list, one is struck by the complexity of even the simplest forms of life. Okay? All right. So why don't we take a pause there and just take a question or two, and then we're going to talk about the issue of homochirality, which I'll explain. Fancy chemical word. So who's got a question about the complexity of early life? Anyone? Take one question.
How did they find out, how did they realize it was that complex?
Because of the nature of the organisms. So they were able to look in the fossil record and they see these cyanobacteria. And they can compare them to cyanobacteria today. And what they notice is they haven't changed a whole lot. And they know today they can grab a hold of what we call blue-green algae, for example, and we can examine them and they can see the complex metabolic pathways that a blue-green algae has to have to do all these things. When you look at it with the naked eye, you just think it's a bunch of muck. But these are complex cells. They're capable of doing photosynthesis and all these metabolic pathways. And in the fossil record, we could see the evidence of them. And even though it was a long, long time ago, they appeared almost exactly the same. So in other words, when they appeared with all this functionality. Otherwise, they wouldn't be a cell. It's a great question.
What would be considered a simple form of life that they were expecting?
Well, a protocell, for example, is what I think you're referring to. A protocell would not have had all those things in it that I described. So a protocell might have had a very simple piece of, I don't know, nucleic acid and maybe some simple proteins, but it would not necessarily have acquired the ability to replicate itself, to grow and develop into new cells, let's say. So it only acquired that later on. That would be a more advanced function. But that's not what we see. What we see is that cells had the ability to replicate and grow from the get-go. There is no evidence for protocells, as it were. So why don't we carry on, and we're going to talk about homochirality. I'm going to introduce the idea to you, and because we've got a little bit of time here. So if you look at the picture that I've put up, you'll see there that something that has a mirror image of itself that's exactly the same. So if you look at on the right-hand side, you can see a glass, okay? And you've got a glass and a mirror. And it doesn't matter which way you look at it, they're exactly the same. You can interchange the one over top of the other. In other words, they're superimposable. You with me? But if you notice on the left, you've got a hand. And if you were to bring your hands up to a mirror, they're not superimposable. In other words, if I take this hand and try and put it over top of this hand, I go, oh, my thumb is where my little finger is, right? So it's not superimposable, okay? And this is what's known as chirality. In other words, it's the handedness of some of the molecules that we see in life. So you guys all know what a molecule is, right? You have an atom, let's say carbon, and then you have 4 different things hanging off of it, right? Well, if those 4 different things are all the same, then you can put the one next to the other and you can superimpose them. But if the 4 different things are different, It depends on where you attach the four different things, whether or not it's superimposable or not. And this is the notion of chirality. So here's a little picture. You can see this molecule here. It's got a carboxyl group and an amide group. They are two different molecules that can't be superimposed. Okay? Now here's the thing about all of the biomolecules that we see in life. And this is something that I think is mind-blowing. Okay? When we look at all of the random processes that take place in chemistry, take A, you put it together with B, and just mix them together and watch it work, they should produce equal mixtures of what we call stereoisomers. And a stereoisomer is basically the left hand and the right hand, okay? But they should produce equal amounts. But as we look at all of life's biomolecules, those that have this feature of being handedness, they're all just one hand. They're all what we call left-handed, for lack of a better argument. It's just left-handed molecules. So all your DNA, your nucleic acids, all your sugars and things that exist in cells are all just one. But if everything's happening through random chemical processes, you should get equal amounts of right hand and the left hand. But instead we don't. With life molecules, we only see one of those. Now that's amazing. That doesn't make any sense whatsoever from a natural process. Okay? So let's leave it there, and we're going to pick it up at the next session. We're going to carry on to talk a little bit about what we call the chicken and egg challenge. All right, so thanks very much.